Here we go. And here we are. I've always said I think of this course as being like climbing a mountain. We've had some really good rambles in the first part of the course. From genetics to transcription and translation is kind of a hard climb for some people. Once we're at the top, there's a lot more that we can see. And there's so many directions we could go in from here, and there's just not going to be time to cover everything that I wish we could. But I'm going to point out some things that we can now see from the top of the mountain. And I'll point them out as I'll pick some that I think are relevant and cover as much as I can before we have to start heading downhill. So that's a tobacco plant. And for at least a couple centuries, tobacco farmers knew about a disease that could hit the crop. Affected tobacco plants develop these light patches on the leaves that are irregular in shape. And because you have these, light patches on a dark leaf, the disease was called tobacco mosaic disease. And a couple of people started studying this, Ivanovsky in Russia and a Dutch scientist named Martinus Beijerinck. And they both found that, first of all, tobacco mosaic disease was caused by some kind of infectious agent, because you could take an infected leaf and grind it up and paint the leaf juice on a healthy plant, and the healthy plant would get the disease. So this is not because there aren't nutrients in the soil or anything like that. This is behaving like an infectious disease. The problem was that they could filter the juice through a filter that was so fine that no bacteria could get through. You could filter this juice and you wouldn't find any bacteria in it if you examined it under the microscope. You couldn't grow any bacteria on culture plates. And yet, even the filtered juice would still make the plant sick. So whatever's doing it is something that's smaller than any bacterium, smaller than the smallest holes in the filter. Beijerinck borrowed an old Latin word for poison and named this agent a filterable virus. Virus in Latin originally meant a poison. Wasn't until the 1930s that we could see what this filterable virus was. That's when German scientists, I believe, invented a much more powerful type of microscope called the electron microscope. I've mentioned it before. I've shown you some pictures taken through it. We have one here at UCA. They've got a couple of different types at UALR. And the electron microscope is what let us see the tobacco mosaic virus for the first time. Turns out it's only a third of a micron long. Remember that a micron is a millionth of a meter. The absolutely smallest objects you could possibly see with a light microscope under the best conditions, with the best possible, highest quality scope ever made, is only about half a micron. So this thing is smaller than the most powerful light microscopes can make out. It's not made of cells. It doesn't have cell membrane. It doesn't, have organelles or anything like that. And it doesn't have any metabolism. If you take this thing out of tobacco plants and just let it sit in a vial, it does nothing. It doesn't breathe. It doesn't excrete. It doesn't take in molecules. It doesn't build up new molecules or break down old ones. It just sits there. When it's outside of a living system, it is about as lively as a pebble. In fact, you could actually make crystals out of the stuff. So what the heck is this thing? Viruses are pieces of either DNA or RNA surrounded by a protein coat called a capsid. The capsid is made of one or a few types of protein that pack together. And capsid proteins, when a virus is forming, will self-assemble. All right, jump to slide there. In the closest analogy I can think of is the way that snowflakes form in the air. There are just natural, regular ways that water molecules will pack together when you cool them and let them start sticking to each other. And that's how you get orderly looking structures like snowflakes or frost on the window or things like that. Viruses form in an analogous way. They're not made of cells. They're not cells. If you take them out of a living system, they do absolutely nothing. But they can, in fact, take over a living cell and replicate inside a living cell. So they kind of straddle the boundary between alive and not alive. They can't reproduce on their own and they don't have metabolism, but they are made of DNA or RNA and proteins. They can reproduce, they can evolve. And so this would be a good time for you to go back and look at those features of life that we talked about in a very early lecture back in January, I think. And you'll see that viruses don't check all of those boxes. And that's okay. Sometimes when we come up with nice, neat definitions, life has a way of giving us the finger. So there's a virus capsid. This one happens to be one that people were very concerned about a few years ago. It's the Zika virus. This is the one that can cause damage to fetuses in a pregnant woman. And you're looking at a model of the capsid, and each of those colored areas is a different protein. And you can see how they pack together in this very regular, very geometric way. Sorry, I'm just checking the chat and letting a couple of people in if I'm delaying. So the capsid of a virus is often very regular like this, and the proteins are shaped in such a way that if you put them into a cell, they can spontaneously self-assemble to make capsids like this. Inside the capsid, there is genetic material. It can be DNA or RNA, depending on what type of virus it is. This is a model of a piece of a tobacco mosaic virus. The proteins are packed together in yellow, and they're on the outside. And there's an empty space on the inside with RNA coiled up inside it. And this, by the way, was Roz Franklin's other major accomplishment. Aside from working out the size and helical shape of DNA, she went on to some other projects, and one of them was using X-ray crystallography to work out the basic structure of tobacco mosaic virus. So if we go back a couple there, each tobacco mosaic virus is that long rod. And you can maybe just barely see, if you enlarge this picture and maybe take some drugs, that there is a very thin little channel in the center of that capsid. So there's one like running right down the middle there. You can sort of see it. And, there you can sort of see it. That channel right there is where the virus's RNA fits in. All right, so there's a close-up of that. And yeah, there's that channel running right down the middle of a tobacco mosaic virus with RNA stuck in it. This is one that infects bacteria with a very complex capsid that actually looks kind of like the moon lander. This type is called a bacteriophage. On the right, that's a computer graphic of what it looks like. That white stuff kind of balled up there in the head is that's DNA. The capsule is, well, part of it is this head made up of proteins. There's a neck right here and a little base plate and tail fibers right there. So that's a computer graphic. And over here, this is what they look like visualized with the electron microscope using a special technique called shadow casting. That's one. This is pictured with the electron microscope as well. That's a bacteriophage that has latched onto a host bacterium and has been caught in the act of injecting its own DNA directly into the host. And what will happen now is this. Once the bacteriophage injects its own DNA into the host bacterial cell will start transcribing that DNA, making messenger RNAs, and it will start translating those messenger RNAs and making proteins. The proteins will self-assemble into new capsids. Sorry, it was telling me my internet connection was unstable, but in case you missed it, when the bacteriophage injects its own DNA into the bacterial cell, the bacterium starts transcribing that virus DNA instead of its own DNA. And the messenger RNAs get translated into virus proteins instead of the proteins the bacteria need. Those virus proteins self-assemble into new capsids. They pick up new copies of the virus's DNA because the virus DNA gets replicated by the bacterium. So new viruses assemble inside the host and eventually burst out. And this is what is happening here. You're looking at a slice through a bacterial cell taken with a transmission electron microscope. All of those black dots inside the cell are virus particles that are forming. And then you can see a whole bunch of virus particles that are outside the bacterial cell. Those have burst out and they can drift. They'll drift on until they find other bacteria to infect. And once they have all burst out, the host bacterium is dead. The virus basically takes all of the bacterium's machinery of DNA replication, transcription, and translation and uses it to make more viruses. And this is why certain computer programs are called viruses as well. If you get a computer virus, it will take your computer resources and use them to copy itself and then send copies to everyone in, you know, your email address book or your Facebook feed or whatever it might be. A computer virus uses your computer to make and spread more copies of itself. Biological viruses do much the same thing. They use a cell's DNA replication, transcription, and translation abilities to make more copies of themselves and then go on to infect more cells. In fact, bacteriophage viruses like this one have been used to treat bacterial infections. The idea being that if you have bacteria in your body making you sick, you can attack them with bacteriophage viruses, which will kill off the bacteria but not affect your own cells. Bacteriophage cannot infect you. This was done back in the Soviet Union in the 30s. It can work. It was kind of forgotten with the discovery of antibiotics, but there's interest in trying to, you know, redevelop this therapy and, you know, get it medically workable. because it has worked on people in the past. The same basic principles apply to viruses infecting eukaryotic cells, including yourself. If you have a virus infection, which could be, anything from the common cold to COVID-19 to full-blown HIV, whatever it is, the viruses enter your cells and they take over your cells and force the cells to create more copies of the virus rather than the proteins and other things that you need. And then they'll come bursting out of your cells and spread to other cells. There are some viruses that will actually take pieces of the host cell membrane when they burst out. These are pictures of a human herpes virus. One type of herpes virus causes genital herpes. Other varieties cause cold sores, and I don't happen to know which one this is, but the capsid is right there. At this level, you can even see the individual protein molecules that are packed together to make up that capsid. But surrounding the capsid, you have this, and this is a torn off piece of host cell membrane. When the viruses burst out of a human cell, they ripped off little pieces of the membrane and took them with them. That enables the virus in part to evade the host immune system. This is part of the reason why herpes is not very easy to treat. There again, you've got the capsid of a herpes virus right there, surrounded by a ripped off piece of the dead host cell membrane. Kind of like Buffalo Bob in Silence of the Lambs. Ew, sorry. Right, checking the chat to see, okay, there's somebody checking in. Okay, and here's coronavirus. It is wrapped in cell membrane as well. The actual capsid is going to be inside here somewhere. Surrounding it is host cell membrane, but the virus, as it's assembling, also produces special proteins called spike proteins. You can see them, they're the things that are sticking out like this. Those are the spike proteins, also called fusion proteins, and they stick out like the spikes on a crown. This is why it's called a coronavirus. Corona is Latin for crown. And these viruses really do look exactly like crowns if you're on drugs. So that's an electron microscope image at the top, and there's a computer graphic reconstruction down there at the bottom. And that is a slice through a eukaryotic cell that is infected with a coronavirus. I don't think this is COVID-19. Viruses, you don't really classify under the system that Linnaeus used. Viruses don't really have genus and species names, and they're not grouped in quite the same way, but we do talk about families of virus. And there's a whole family of coronaviruses, one of which was big news maybe 5 years ago, called SARS. That was what? SARS. The RS stood for respiratory syndrome. Off the bat, I could always look it up. I've got a cell phone right here. What the heck? Yeah, SARS, severe acute respiratory syndrome, appeared in 2002. Okay, so that was more like 20 years ago. It's all right. It's been a year of quarantine. Time no longer has any meaning. And I think that's a cell infected with the SARS virus, but that's a coronavirus. The COVID-19 virus would look virtually identical to this. And you can see various membranes of the cell itself. This right here, this is going to be the, hang on, whoops. Yeah, that's the nuclear membrane right there. So you're seeing part of the nucleus. Here's part of the cytoplasm, and all this space right here is jam-packed full of coronaviruses forming. And eventually the coronaviruses will go bursting out of the cell, taking some cell membrane with it, and go on to infect other cells. A little more about protein structure. This is a human protein called ACE2. The way it's been diagrammed, each of these things that look like rods, that's actually an alpha helix. So there's an alpha helix, there's an alpha helix. You're probably definitely sick of talking about alpha helices, but that's what they are. It's made of 4 subunits in the primary structure, right? 1, 2, 3, 4. They're different colors. And it's a cell surface protein because cells have proteins on their surfaces that actually go through the membrane. There's a part that sticks out and a part that sticks in. And so the cell membrane would actually be more or less at this level. And the part that sticks out can bind to other proteins or other molecules on the outside and then change shape and cause something to happen on the inside. So you could think of these as being little antennas that enable cells to respond to the environments that they're in. This particular one, ACE2, enables a cell to respond to a hormone that regulates blood pressure called angiotensin. A molecule of the hormone will bind to this and cause the cell to alter its shape. And that will set off events going on inside the cell that involve lots of other proteins. So this is how cells in your body can respond to a hormone that I believe your kidneys put out that regulates your blood pressure. The reason that matters is that the spike proteins of the COVID virus, SARS-CoV-2, are shown in pink. This thing up here at the top is a COVID virus. And these things that look like, these pink things right there, those are the spike proteins. And they have just the right shape that they can bind to these ACE2 proteins that the cell makes. And once they've done that, the virus is essentially able to recognize, hey, this is a cell I can infect. And the cell will take the virus in. It will allow the virus to enter. And then the virus will start doing that thing that it does. And by the way, off here in the background, this thing right here, this mound, that is a new COVID virus caught in the act of bursting out of an infected cell. So there's a new COVID virus being formed, while here in the foreground, this is a COVID virus locking on to one of the cell's protein receptors and getting in that way. That protein has a very important function. You can't do without it, but it's the weakness by which the virus gets into your cells. A little bit about a type of viruses called retroviruses. They're called retroviruses because they own lava lamps and listen to a lot of Doobie Brothers albums and wear bell bottoms and paisley shirts that they unbutton all the way down to the navel and gold medallions. Actually, I'm kidding. Retroviruses are called that because they contain RNA instead of DNA. Some of that RNA codes for an enzyme called reverse transcriptase. And this is weird. Reverse transcriptase will take the virus's RNA and make a complementary DNA strand. Now, at the end of our last lecture, I mentioned something called the central dogma of molecular biology, which states that information is stored in DNA. When the information is transcribed, it's used to make RNA. And then when the RNA is translated, it's used to assemble a protein. This is one of a very few times when that actually runs backwards. You usually use DNA to make an RNA strand. This is maybe the only time when you use RNA to make a DNA strand. It's called reverse transcription, and it's done by an enzyme called reverse transcriptase. That new DNA gets spliced right into the host cell's DNA, and then it gets transcribed, and the messenger RNA gets translated, and that makes new retroviruses inside the host cell. This was first discovered in, I believe it was some obscure chicken virus or something like that. There is one retrovirus can cause a form of leukemia, but the famous retrovirus that does this is HIV, human immunodeficiency virus. Here's why HIV is bad for you. You defend against viruses using a population of white blood cells. Almost all of the cells in your blood are just oxygen carriers. They're your red blood cells. But you have white blood cells in your bloodstream that can leave your blood and patrol your entire body and chase down and kill invading bacteria. In fact, you can produce so many white blood cells that if you've got a bacterial infection, your body will send so many white cells to fight that infection that they form pus. Pus is basically a huge mass of dead and dying bacteria and white blood cells. And there's one particular type of white cell called cytotoxic T cells. And in this photo, the original photo's in black and white, but somebody with Photoshop has colored the cytotoxic T cell orange. And what it's doing is crawling right up to a cancer cell, which is in purple, and destroying it. So cytotoxic T cells defend you against cancer. They also defend you against virus-infected cells. They'll destroy a virus-infected cell before it can release viruses. So they're an important part of your immune response to viruses. The problem with HIV is that the cells that it attacks happen to be these cytotoxic T cells. The virus infects a part of the very immune system that is supposed to defend you against viruses. Much like, imagine a criminal gang infiltrating a police department. HIV works in much the same way. So HIV, by the way, also wraps itself in cell membrane, and it attacks, I misspoke a little bit, it doesn't attack your CD8 cells, but it attacks immune system cells called CD4 cells, or so-called helper T cells, which help the CD8s. So HIV damages the body's defenses against infections. And if you have full-blown AIDS, acquired immune deficiency syndrome, you don't die of the virus. You start dying of infections that most people would shrug off right away.
Because it's weakening
so
your
you
immune
can,
system.
so yes, you can get, there's a particular type of yeast called Pneumocystis jirovecii that most people would just shrug off without even knowing they had it. But if you have HIV infection, your body cannot fight off pneumocystis anymore. So you get this nasty fungal pneumonia. There's particular types of cancers that you can start getting. One called Kaposi's sarcoma that doesn't bother, very rare in the general population, but you're prone to get it if you've got full-blown AIDS. There's various other things that you're prone to getting that will ultimately kill you if you've got full-on AIDS. It's because your immune system's been knocked down by HIV. So, yeah, many of you don't. Now, I'll get to that in a minute. Antibiotics don't work against viruses, by the way. Some people demand that they get antibiotics whenever they get something like the common cold. Well, the common cold is caused by viruses, not even one single virus. There's probably hundreds of different viruses that all cause the common cold. And antibiotics do nothing because antibiotics go after bacteria. Antibiotics interfere with bacterial metabolism. Viruses don't have metabolism. Same thing. If you get the flu, don't demand that your doctor prescribe antibiotics because it will do absolutely no good. It's like trying to use a submarine to shoot down bombers. There are some antivirals that we do have. This is a protein. Notice lots and lots of alpha helices, all those little telephone cord spirals. Interferon stimulates the immune system to step up its game against a virus infection. And then here's some more antivirals. Up here at the top, that's a real nucleotide. It's guanidine. Right here, you've got the phosphate. Not everybody got these questions on the exam, and you're probably sick and tired of hearing about it, but that's the phosphate. There's the sugar, right? That's deoxyribose. And right here, we're all about the base. And now you're going to be earwormed with that. So that's the base, guanidine phosphate. There's, or guanine. And then there's the sugar and the phosphate. This is a drug called acyclovir. And right here, it's got guanine. But you'll notice that the sugar is mostly missing. This is a fake nucleotide. And if you feed a cell full of acyclovir, it will think it's a nucleotide and it will try to incorporate it in making DNA or RNA, but it won't be able to because the sugar is faulty. This group right here, this thing that says OH, that's an oxygen and hydrogen atom together. That's what is supposed to link with the next phosphate. And then this phosphate will link with another sugar right up here. So what holds those sides of the twisted ladder together is bonds between the phosphate, and this OH group on the sugar. Here there is no OH group, so a cell will try to use this to make DNA or RNA, but it's faulty. And if it gets any of this in its own DNA, it can't make the DNA grow any larger. That blocks DNA replication and it blocks transcription. Now, that's poisonous for the cell, but it's even more so if the cell is infected with viruses. It stops viruses from being made. And that's why acyclovir is used to treat herpes. I don't believe it's an absolute cure, but it can knock herpes infections down very effectively. The same is true for galidesivir, which has a base right here. That's adenine. There's your sugar. And there's the phosphate. So that's adenosine monophosphate. Galidesivir, it doesn't have a phosphate, but your body can always add one. That's not really important. it's got the base adenine, and the sugar is wonky because right here it's got a nitrogen atom in it. That gives that sugar the wrong shape. So galidesivir also looks like it's a nucleotide, but it isn't really. And if you dose a cell with galidesivir, it will also interfere with DNA replication and or RNA transcription. Galidesivir was developed to treat a virus infection, hepatitis C. It looks to be effective against things like the Ebola virus, which was big news a few years ago. And I looked up the company that's making it. I think they're called BioCryst, is testing it against coronavirus. And as of December of 2020, the last news I could get is that early clinical trials were promising. They have found that it is very safe. They found that it does seem to lower what we call the viral load, lower the total number of viruses in a person that's been infected with the coronavirus. I don't think it's got official approval as yet, but it does seem to work, but more testing is still going to have to be done. So that's how you can treat viruses using fake nucleotides. One last one, dideoxyinosidine. That's an actual nucleotide with our friends, the phosphate, the sugar, and there's the base. That's adenine. And then the drug Videx uses a different base. It's actually called inositol right there. And notice that the sugar right here is missing that little OH right there, that group of an oxygen and a hydrogen atom, which means this is not ribose, it's not deoxyribose, it's dideoxyribose. It's lost two oxygens right there. And remember that that oxygen right there is what will link to the next phosphate on the chain of nucleotides that makes up DNA that makes up DNA. If you don't have that oxygen atom right there, you can't link this to the next nucleotide any more than a train car without a hitch can be linked to the next car, or any more that a pickup without a ball hitch can be linked up to a trailer. So if you dose a cell that's copying virus DNA or RNA with Videx, you block strands from extending further. And I'm old enough to remember when news about HIV came out. Many of you don't even remember this, but it went through the gay community like the Grim Reaper. There were so many men who lost all of their friends. There was not a great deal that doctors could do, and an awful lot of really bad misinformation being spread about HIV. Shoot, somebody in my church youth group informed us one evening that we were all going to die because mosquitoes were going to spread HIV from one person to the other as they bid us to drink blood. And in South Louisiana, there's a hell of a lot of mosquitoes. Fortunately, that turned out not to be true. But yeah, there was a great deal of fear about this back in the 80s. There was all kinds of politics going on. I probably shouldn't rehash too much of this. But what HIV has become is it's no longer a death sentence as long as you can get the drugs needed to manage it. It's not something you should try to get. So, keep using those condoms and everything. Be careful. But it's no longer something that means you're going to die within a year horribly and probably be abandoned by your family or something like that. That used to happen all the time. HIV patients now take an entire cocktail of drugs that block transcription other drugs that interfere with those fusion proteins, and others that block the virus's assembly or that block the viruses from breaking out of the host cell. So you treat HIV by hitting it with multiple drugs, each of which works in a different way. And all of these strategies can be used to treat other viruses. So there are drugs that block flu viruses from breaking out that can provide relief in severe flu infections. And I hope you see that all of this stuff that we've been pushing through is of very direct relevance in understanding public health today, because there's lots of viruses that cause disease, some of it very severe. And we can use what we know about transcription, translation, protein shape, and everything else to work out effective treatments. And I will go ahead and stop there because I've gone a minute over. So we will now stop the recording. That'll be an extra $5 for each of you for that extra minute. Just kidding.